In the eukaryotic cell cycle, we go through a number of different phases. The first is interphase. This is essentially the rest and growth phase. We also look at replication of the DNA only occurring in this phase. So G1 is growth. This is the start of the entire cycle. S is synthesis of DNA, or replication of the DNA only. G2, we look at more growth. If we're going to divide a cell in half, it has to get bigger first. So the cell grows and grows and grows, and then we go through mitosis, or cell division. Mitosis is the division of the nucleus. Cytokinesis is division of the cytoplasm, or the cell itself. So in this phase that we start, so if this is our start point, we're going to grow. We're going to go through DNA synthesis or replication. We're going to grow again, and then we're going to divide. So mitosis is division of the nucleus and of the cell itself, as we mentioned. On a 24-hour cycle, which is roughly what it takes for your cell to go through an entire cycle of cell division, or the entire cell cycle, as we refer to it, this whole process of division takes about 1 hour. The rest takes about 23 hours, depending on the cell. Now, this might seem like a long time. It's not particularly a long time, because you've got to remember, every cell in your body that is active is going through this every single day. Now, when we talked about prokaryotes, that binary fission only takes about 20 minutes. So the complexity of the cells with growing and organelles and having to go through a lot more DNA replication and processing takes much, much, much longer than we saw in the prokaryotes. Mitosis has 6 major stages that we're going to look at: prophase, prometaphase, metaphase, anaphase, telophase, and cytokinesis. One of these you already know. Cytokinesis, we said, is cell division. We're going to need a couple of players in this, other than the cell itself. We're going to have to produce mitotic spindles. We've talked about these before when we talked about cytoskeleton. They're made of microtubules, and they're produced by something known as the centrosomes. You're going to have to be very, very careful with these C words in this section. There are many of them: chromosomes, chromatin, chromatid, centrosomes, centromeres. You're going to need to make yourself a list to kind of give yourself definitions for what is going on. These mitotic spindles are required. They're actually going to be the movers of the chromosome. So in interphase, this growth, synthesis of DNA, and growth that we just looked at starts us into prophase. You see our chromosomes are in this X form. They're in their sister chromatid replicated state. They better be. We went through DNA synthesis, so they have to be replicated at this point. They're held together by that centromere that we mentioned. Here are centrosomes beginning to build our mitotic spindles. And this phase I remember as plain to see. P, P. And what I mean is I can clearly see the chromosomes inside my nucleus. So remember, the purple is the nucleus. In prometaphase, we're going to break down the nucleus. You see the nucleus has disappeared inside the cell. And then the spindle fibers, or these microtubules, are going to reach out and grab on to the centromere in the middle of the chromosomes. And you see they're coming from both sides. And the idea is we want to now attach to separate the sister chromatids so that we can put them into two new cells. Metaphase, M is for middle. All of the chromosomes are lined up in the middle along something known as the metaphase plate. And you see they're all quite organized, sitting here in the middle, oriented all the same way, so that in anaphase we can pull apart, A for apart, pull apart the sister chromatids. And you see, as soon as they're no longer connected, we now call them chromosomes. We only talk about them being chromatids when they are connected together at that centromere. In telophase, two nuclei. So the cells are going to come apart right here in the middle through cytokinesis, and you see these little purple spots. Those are pieces of the nuclear envelope that are reforming. Or that nuclear membrane we talked about before. So they're going to form two new nuclei, and when that occurs, this cleavage furrow, this split in the middle, is going to squeeze together and actually literally pinch these two cells apart as the plasma membrane touches in the middle. So this is what it looks like as it starts to fold in during this process of squeezing in and then separating these two cells. It actually occurs like a string on the hood of your hoodie. If you pull it really tight, you know how it pinches together? That's exactly what's happening. There's a contracting ring of microfilaments that are going to pinch together and actually pinch the plasma membrane together so that the cell will then become two separate cells in the end. And the whole idea is to build these two daughter cells, right, with two nuclei, and these are perfect copies because we did mitosis. So that produces perfect copies of our cells. Now, this process works exactly the same in plants up to this point. You go back to prophase, prometaphase, metaphase, anaphase, telophase. All are the same until cytokinesis. What do you think the problem is with plants in cytokinesis? Remember, they have a cell wall. This is a problem. Cell wall is not going to pinch shut. The plasma membrane on the inside will, just like in animals. But the problem is this cell wall. So we actually have to grow something known as a cell plate. The cell plate will actually start building from the middle and eventually divide this cell in half, so that each new cell will have cell wall and plasma membrane. Now, in this process, we look at this cell. These two new daughter cells are going to be smaller than the original parent cell because this cell wall has kept them from growing too much larger during this process, and we kind of keep dividing until we get these smaller cells. Now, that doesn't mean that plant cells will become infinitely small. They will take time to grow. So now these two daughter cells will go into interphase, just like animal cells, and they will go through growth, synthesis, and growth. They just seem to get much smaller during this division because of that cell wall. So just keep that in mind.